this is a great opportunity to have not just some theoretical work, but some actual on the ground, getting it done, domestication work that Lee has been leading at the Land Institute there in Kansas in the US. And so I'm sure Lee will introduce this, but the Land Institute and really the premise and the focus of the institution is on developing perennial grain crops for this combination of ecosystem services, sustainable agriculture, and then having not just a perennial, but a grain crop that can be utilized for human consumption. And so Lee is a wheatgrass breeder, this intermediate wheatgrass. And so he'll talk about that today. We had the great fortune of being teamed up with Lee, boy, almost a decade ago now, probably. We were doing a lot of work in genomic selection in wheat and seeing how this could accelerate the breeding process in wheat. And Lee jumped all over this, and it's really been a beautiful implementation and a lot of success to speed up the domestication and the improvement of this really unique crop. And so, like I said, I think this is we have this opportunities to hear some of these really great examples on these domestication programs that are really happening and actually having product and domesticated species coming out of them. So it's bright and shiny early for Lee. I think 6 a.m. We didn't take the time change into account when we scheduled all of these things. So it's an hour earlier than it would have been 2 weeks ago. So anyway, thanks, Lee, for joining us super early. I guess, like you said, it's still less onerous than the jet lag of being here. Yep. So we'll turn that over to you and go ahead.
Thanks, Jesse. And the time frame we're talking about today?
What's up?
What's our time schedule today?
Oh, yeah. So we have an hour put aside here for the seminar, and then we'll have question time after that.
Right.
So we have maybe, just shy of an hour for seminar, followed with questions. And then after that, we'll have about an hour put aside for seminar and discussion, for journal club and discussion of this paper with the students and others that come.
Okay. Sounds good. I'm going to cover a lot of territory quickly. I've been at the Land Institute for about 20 years. And in that time, I worked on perennial wheat initially by crossing with wild perennial relatives of wheat. And I had a side project of directly domesticating a wild relative, intermediate wheatgrass, Thinopyrum intermedium. It's now on the market in a very small way. And because retailers didn't want to put Thinopyrum intermedium on a package, we made up a new name. We call it Kernza, Kernza. So you'll find some of the papers and some of the work. People are using that name just because it's easier to talk about. So if I ever accidentally say that, I'll probably mostly say wheatgrass today to talk about the species, but that's what we're talking about. So I had imagined that this project would take probably at least 50 years to bring it to something that would be useful to farmers. It started going faster than I thought. So in 2010, I suggested that maybe by 2020, we could get to the point where farmers would be growing it if I worked on it full time. So I moved full time to this. And we since hired another breeder that takes over the perennial wheat by wide hybridization project, which is another story all in itself and also has exciting results in the last few years. I'm going to focus just on the domestication though. So today I want to talk to you about not just our recent work, but why are we doing it? So it's a really difficult project, a long-term project, and it's not worth undertaking unless there's a good reason to do it. So I want to talk about why we're doing it and why we picked this species and how we've been going about it and how we think we can go faster in the future. So I grew up on a farm in north central US that grew conventional crops, corn and soybeans. This is actually my dad and the tractor. I spent thousands of hours on tractors like this as a kid, tilling the soil. We were low tillage, but we still did mostly conventional tillage in the sense of we weren't doing no-till at that time. And spent a lot of time thinking about what the ramifications of all of this turning under of crop residues and making black soil and planting annual grains every year might be having for our soils and for the world as we do this all over the place. And that we know historically the results haven't been good. This is just a photo of the Lost Plateau in China, where agriculture was practiced for hundreds to 1,000 years, and the results were in a landscape where they couldn't grow crops anymore. And this has kind of been repeated throughout history. I've just got a quote from Lyle here talking about how civilizations have fallen due to the undermining of their soils. Incidentally, I see Lyle just received the World Food Prize this year, and so he knows some stuff about soil and what's happened. And the founder of the Land Institute, Wes Jackson, was basically moved by this idea of we can't undermine the soil that's essential for the production of food, and we have to really our work here at the Land Institute started at the very beginning of the term sustainable agriculture. Some people credit with Wes Jackson for coining that term. So the idea arising in the 70s and in our founding here of we have to find a way to produce food that doesn't undermine our ability to do it in the future. And Wes was really taken by the prairies and the thought that what is it that's different about our grain production systems that make them deplete soil versus perennial grasslands of the world that build soil. And I want to kind of talk about that difference just in the ecological context. So it's not often that we see land being cleared in nature. So an example would be a volcano. This happened in my childhood, the eruption of Mount St. Helens in the US that cleared all this land. And I just remember magazines having pictures every 5 years or so of what happened. And slowly small plants started coming back in, and then eventually trees come back in. But this is a rare thing in nature. Usually a disturbance that clears all vegetation very, very seldom happens in nature. And in agriculture, on the other hand, we're doing it basically on a yearly basis. We remove all above ground biomass and often kill the below ground biomass as well. So in order to plant our grain crops, we push the ecological reset button every year. And that puts us in a situation where soil, nutrients, and water are all leaving that system in very high amounts and degrading the soil system. And tillage goes even farther of oxidizing soil organic matter and depleting soil quality. So the dream is, could we get back to something that's more like the grassland above here and have a way to eat that directly? So although it's very sustainable for us to raise cattle on grasslands, the productivity of that system is pretty low. And if we could only eat those plants directly, we could have that same sort of sustainability that we have of natural grasslands while also being able to eat it. Trouble is, all these plants that are native in our part of the world produce very, very little edible food for humans. So we're trying to coax these plants into beginning to produce more grain that we can eat. And the problems in agriculture have not necessarily been solved in recent years, even through the development of no-till has been a vast improvement, but we still see catastrophic erosion happening around the world. This is Iowa, the center of corn production in the US. And there's days where there's been heavy rainfall in Iowa where the average soil loss has been 10 to 20 tons in one heavy rainfall event in some counties in Iowa. So we still have very unsustainable rates for the long term, even with our best methods of annual grain production. This is a somewhat of a complicated graph that my colleague Tim Cruz made up. in a recent publication. I don't know if I want to go into all of it, but basically it tells the story of succession over time as land is disturbed. So if we look at the top, this is soil organic matter, and this might be a prairie or a grassland. And as this was plowed for use in annual grain production, there was a steady decline over about 20 years, 30 years. The soil carbon or soil organic matter declined to about half of what it was initially in the part of the world that we live in here. And we ended up at kind of this new steady state, this early successional state where I mentioned we're hitting the ecological reset button all the time. And if we know if we replant that to grasslands, there'll be a steady increase in soil organic matter again over time. And Tim's hypothesis here is that if we were to have a perennial grain cropping system, we imagine the same sort of recovery. Maybe we don't get to quite the same level, but a steady increase and get to a new steady state where we have very enhanced soil quality again. Jesse, can you see my pointer when I move it on the screen?
Yeah, we see
Okay.
it just fine.
All right, thanks. So other things follow a similar trend. This is actually net ecosystem production. So it's basically that movement of soil carbon over time in or out of the system. Follows a curve of where there's this decline and initially, and then as it's tilled under, and then building up to this steady state. And eventually you want to get to a situation where basically there's the same amount of carbon moving in and out of the system. Plant removal, plant available from soil organic matter. So there was this big flush of nutrients. So as we plowed the prairies, there was no fertilizers here in the late 1800s and early 1900s, but wheat yields were amazing for several decades. And that's this flush of nutrients that came out. And then there was a crisis in the 20s as yields crashed around here. And as we discovered fertilizers, we were able to make up for that with soil fertilizers that's shown by the blue line. And Tim's envisioning here is that eventually we get to, if we were to bring in a restoration of perennial plants to that system, we would get back to where a similar situation in terms of a steady state for soil organic matter providing nitrogen, but we still have to have some sort of fertilizer or legumes used to provide nitrogen, but much less, because 50% of what is applied to annual grain crops worldwide is lost to the outside the ecosystem. So it goes into water or air. And we see that with perennial crops, we retain 90 to 99% of what's applied actually stays in the system instead of slipping out. So we can get by with about half as much and still get the same yield off that land. And similar kind of trend here and this explains what's going on there. So gaseous or leaching losses are really high in these disturbed annual grain production systems. And our vision with the perennial system is to get back to a highly conservative system. That's going to happen through deep root systems that are in the soil, not allowing those nutrients and water to slip away, using it before it gets out of the reach of the roots. And just inevitably, when we till or kill everything and have to start from a single small seed with a very short root, there's this, we can follow the pulse of nitrogen fertilizers that escapes these root systems and goes into the water or gets lost to the atmosphere. All of that soil carbon does a lot of good things in terms of the biota and makes a healthier soil, and eventually would, as we restore this, will increase the productivity of the soil again. I mentioned nitrogen. It's not only costing farmers, but it's damaging ocean systems around the world as the nitrogen leaches into those systems and causes hypoxic zones as oxygen is used up, and all the larger life forms have to leave or die from those systems. And it's a global problem. Basically, wherever you see intensive agriculture is being done, nitrogen leaches out of those systems and produces these so-called dead zones in aquatic systems. We have some nice data. As I mentioned, the reduction by 90 to 99% of leaching. This is an organic system with an annual and perennial, or in a conventional system. Similar trends are seen. So once we establish the crop after a couple of years, the amount of nitrogen leaching out of it with a big root system is very close to zero. So we feel there's the payoff for perennial grain crops is worth the large and somewhat risky investment. We don't know if any one of these projects is going to succeed when we start it, and it's going to take a decade or two to know how it goes. But it's worth trying a bunch of them, we think. So why this crop, intermediate wheatgrass? The work on this species has quite a long history in the USA. Started with in the 1930s. These crops were introduced into the United States, mostly for use as a forage. And so it came from close to the part of the world where you all are right now. A lot of it from southern Russia, Afghanistan, Kazakhstan, Turkey, and maybe even farther into Europe. But most of the collections come from those regions, and brought to here as a forage crop for the western part of the U.S., where it's drier and higher elevation. So several forage varieties released over time. Then there was, in the 1980s, the Rodale Institute, which is an organic agriculture research institute, started in the 70s, 80s. Got inspired by the work of the Land Institute here to start trying to develop a perennial grain crop. So they looked at 100 perennial cool season grasses and chose this one as the best candidate based on the size of the seed, how much seed is produced, the ability to mechanically harvest it. And their work resulted in studying the various collections that were available, and then it was passed on to a government organization called the Big Flats Plant Material Center in New York. And they did 2 rounds of selection, just conventional selection. They were fairly small populations. They only had about 400 plants, and they would pick the best 12 to 14 plants each cycle. So they did 2 cycles of selection out of only 400 plants, but did very intensive 5-year evaluation of those plants to make their selections. I obtained those when I started here in 2001, and I evaluated what they'd created against all of the available collections, which was about 450, and nothing was close to as good as what they had in terms of grain yield. So I basically worked within what they had created for first 10 years of very conventional breeding here, and then I'll talk about where I went after that. And here you see it. 2016, that was the first product that appeared in stores being sold in the United States, made from this grain. So the crop is astounding to us in its root biomass, as we had this field with wheat growing side by side with intermediate wheatgrass, and it's very visually apparent. We tried as hard as we could to actually wash this away and find some wheat roots, but the wheat root biomass is much lower, maybe 5 to 10 times as much biomass on this side, and going all the way down to 2.5 meters. And it's that root system that's driving these ecosystem functions that's worthwhile looking at. You can see here that a graph of the total root biomass under some different systems. The annual is wheat, and the intermediate wheatgrass is the perennial here. So it's not only that how much it has, but also the fact that it's there all year round. So it's that time of the year when there's no roots in the system that the annual cropping system is most vulnerable. Wheat in our part of the world is winter wheat, so it's actually there a fair bit of the year. But the summer period, when we have a lot of rainfall happening, there's just a lot of opportunity for leaching losses out of the system. There's a lot of sunlight also that's falling on that ground, and it's basically wasted because there's often no plants there if there's no summer crop planted. In the part of our country where we plant corn and soybeans, there's about 3 months of crop growth, and the rest of the year is fallow. And that's the vulnerable period when we want to have these perennial roots there. One other thing we can touch on is soil carbon and climate change and CO2 sequestration. This is just some data that we had from an eddy covariance tower. If I click this, I get it. Yeah, tell us when it is. So I had an older field of intermediate wheatgrass. So we started tracking the net ecosystem carbon accumulation, which was going on at 5 tons per hectare per year initially. And this is still in year 5, 6, 7. And then it began to decline and reached more of a steady state that was around 1 ton per acre per year. So in this part, we think we're still building basically living and recently dead root mass in the system. But the steady state is probably about 1 ton per hectare per year. And this is just tracking that buildup of carbon over time. Yeah, the average here was 3.7, but we think that the steady state is more like 1. So what would that mean, like for a global perennial grain production system, if we were to, oops, I thought I had a different slide here next. This is a similar slide showing the average being 1 ton per hectare per year in a long-term study of 16 years of a Kernza or intermediate wheatgrass field. It was even higher than restoring native vegetation to that system. So how does this fit into the soil, the global carbon cycle? So I have what I call the back of the plant breeder's envelope. I'm not a soil carbon or global biogeochemist in any stretch of the imagination, but we did work out some of these numbers and basically say and try to answer the question we get often is what would happen if all of the annual grain crops of the world were to become perennial production systems. And so with that sort of 1 ton per hectare per year sequestration, that's not going to happen forever. There's kind of the ability to sequester 40 to 88 gigatons, and that will kind of get us back to where we were before we tilled everything up. And as we do that, we can sequester about 1/2 of a gigaton per year of carbon, and that's, you can see that relative to the 3 to 4 that is going up in the atmosphere every year. So it would be a substantial impact, but it's going to take huge numbers of millions and millions of acres of transformation. So it's not a near-term solution, but definitely increasing the percentage of land that's in perennials versus annual crop production could play a significant role in soil carbon sequestration and impacting the amount that's in the atmosphere. So another answer to why this crop, we planted about 12, 15 years ago now, planted a 30-acre field nearby of intermediate wheatgrass under a grant-funded work, and got the farmer to help us and drove his combine over and found that it was actually very easy. We got a whole semi-load of grain off of this. And so using conventional equipment, this crop can be grown quite easily. And the biomass residue, it can be used for cattle feed. It's not a high quality forage, but it's better than wheat straw by a good stretch of by a good margin. So farmers are excited about it, and the dual use is coming in really handy in years when it's drought. Farmers can keep their livestock alive and feed them on the forage, and on a normal year use the forage and sell the grain. The grain itself was very tasty. It can be used to make products similar to wheat. At 20% in raised loaf, it does really well, and bakers really like it, and people who eat it really like it. 100% it can go into noodles, and there's pancakes and waffles. It's really great. And just in the past year or two, there's a website where you can buy the grain now, and I think they're doing a pretty good sales in the last while. I've been buying them to give to presents for people for over the holidays here. It's very expensive. It's very high-end specialty small market, because the amount that's being grown is still just a few thousand acres, and so everything that's done is very small scale and gets expensive. But it's been useful to have this proof of concept to show that yes, we can take it from the field into products that people will buy. Even at larger scales, Cascadian Farm is owned by General Mills, one of the biggest maker of grain-based foods in our country. And they released just a few months ago this Kernza cereal at select stores, but it's going to be moving into more grocery stores here at the beginning of the year. And they're trying to buy hundreds of thousands of pounds of grain to really scale this up. And so it's just a matter of how fast we can get more farms growing it. Things like cookies that don't require the high gluten strength, it works really well. So it's more like a really soft wheat and its ability to be used in products easily. And a raised bread dough, as you see here, it's way better than using, say, corn flour or sorghum flour. It does have gluten forming capacity. It's just nothing like we see with hard wheat flour. But there's a lot of variability for this. I'm not going to get into the details of how we made this, but this is basically high quality wheat, low quality wheat, and it's this portion of the curve here that's the high molecular weights, glutenins that contribute to the dough strength and quality. And these are individual intermediate wheatgrass plants, and we see a lot of them are terrible, but there's some that are really good. And so I actually started a selection program where I was just picking the ones that were the highest amount of this. And just this year, I've gotten to the point where I'm trying to bulk it up to the amount that we can use to make raised loaves from the grain that's come out of this selection program. We'll see if it's had any impact. Basically, at this point, no one cares that much about quality, because we don't have enough to make 100% loaves anyway. If we mix it at 20%, we can sell millions of pounds at 20% before we're running out of places to go with Kernza flour. So it's not a high priority for the program, but I'm just curious how much progress we can make. Basically, there's so much genetic diversity in the species. I think whatever we want to do, we can achieve. The genes are there. It's just a matter of the time and resources to put into the selection. Farmers are very interested. This is just a graph of farm size of farmers that have called us up over a couple of year period saying they want to try to grow it all over the country. And we also get a lot of inquiries from other countries. So how have I been doing it? Basically, the idea is to get reallocate this all of this leaf and stem material into grain. So there's plenty of biomass produced compared to wheat in various parts of the country. We've seen that Kernza will outproduce wheat by biomass by a long way. So it's not like we have to give up anything here in terms of production of the system. We have to change where it's going through breeding. And this is how we got grain yield in our annual grain crop. So we're confident we can follow that path and do it again. So I dove into this just by doing recurrent selection, growing a lot of plants, picking the best ones, and assuming that over time we're going to accumulate the alleles we need to have a high yielding crop. Initially, I started out with about 1,000 plants per cycle. I got up to doing about 25,000 plants per cycle. This field had, so there's plants about on a 1 meter spacing here, about 22,000 plants. And we harvested heads from every one of those, threshed them, weighed the seed and all that, and searched for the outstanding individuals that would have the best traits for mostly seed size, yield, free threshing and shatter resistance were the traits I cared about most. And it was worth looking at a lot of plants because there was rare plants that were really outstanding that helped us to really move the population quickly over time. But this process takes, if you do it well, it's going to take 3 years or more. I pressed it down to just only evaluating on 1 year to get down to 2 years of evaluation, just to turn it around quickly. And we could do that for traits like shattering and seed size that were pretty consistent year to year. Yield's a different thing. You really need to evaluate multiple years, and we weren't doing that because we're mostly interested in those domestication traits. After several years of breeding like that, 10, 12 years, we got plants that were 100% shatter resistant, as you can see by this. Basically what I did is harvest these plants and just smacked them hard on the table. And all the seeds stay in the head, and the wild type plants, all the seeds basically fall off with just a few taps and all the seed is gone. So that's been very doable just through traditional plant breeding. Seed size has really improved over that period as well. This is the wild type on the left, and there's a couple examples of the newer domesticated forms here on the right, with more than doubling of seed size. So I did 5 years, actually did 6 years, but here I have 5 years evaluated of just phenotypic selection. Initially it was 3 years per cycle, and then down to 2 years per cycle, 1 year of evaluation, then intercrossing, and back to the field again. And so seed mass increased steadily, but not quite as fast and relative to the absolute value here as we did for like free threshing ability. So it's really important to farmers to have the seed to come out cleanly instead of being in a hull like the wild wheats were, because of the small seed they're hauling truckloads of air and hulls around. It's very costly and inefficient. So farmers tell me it's an important trait. And so just in 5 cycles, initially, actually, the first 2 cycles, I didn't select this at all, and you can see why it didn't improve. And then I started realizing it was important, and you see what happened. And then grain yield, we were only selecting on yield per head, but this is actually yield of a solid seeded stand in a production type situation. So even though we weren't selecting for yield in that environment, where I was just selecting on yield per head and seed size, those were strongly correlated with the yield in a crop type situation. You see that we've more than doubled the grain yield. You can see here the numbers are not impressive. So we went from 250 to 600 or so in kilograms per hectare, and farmers now are pretty happy if they're getting 500 to 1,000 kilograms per hectare off their fields. But we see no reason that this can't continue, and we hope accelerate this process so we don't have to wait for another couple of decades before we have substantially higher yields. There might be ways to really accelerate this. So yeah, that one of the first things that Jesse and I did was to talk about getting a reference sequence, and ended up getting some funding along the way. That the Joint Genome Institute of the Department of Energy funded the proposal to work on this, which really helped a lot, combined with a lot of funding from the Land Institute. We did it in our genome initially, which based on short reads, and never really came together all that well. It was hard to get a good quality out of that. Did also RNA sequencing, and this work was done at Hudson Alpha in the US. Frustrations with that led to version 3 of the genome, where I was just putting up some of the how it was done and where we ended up in terms of quality with this genome, which is now the one that's being used for the work that's going forward, based on about 34x coverage with PacBio long reads, and got extremely high quality genome out of this. It's a total of 389 contigs. I think with the RNR gene, we're still at thousands of contigs. So much better, and just a small number of scaffolds across the 21 genomes. So this has been a big help, and it gives us a lot of confidence going forward with the work based on the genome from this point. So Jesse mentioned genomic selection. Initially doing phenotypic selection, 3 to 5 years would be appropriate for field evaluation. And so we're trying to pair this with the genomic selection side. And here we're doing 3 to 5 years of evaluation in the field of a training population, but using that to update our model. So every generation I grow about 4,000 genotyped seedlings, and we apply the model, select about 100 that we intermate in the greenhouse, and then return the next year to the greenhouse with 5,000 seedlings, of which we genotype about 4,000. And we've done this for about 4 or 5 generations now of turning around 1 year cycles. Over the time, our model has improved a fair bit, because initially we only had 2 years of data from one field of about 4,000 plants that were genotyped and phenotyped. That's where we started with the model, and then it's just built with thousands more plants and data points every year. So we now have it based on a model based on 2 locations, many years, and thousands of plants. This is what it looks like in the greenhouse. Just a little before this point, I would have removed the leaf tissues, and when I do that, I put a barcode label on every one as I pull it out to make sure we're not screwing up the plants. And some of them go to the field in the fall that aren't selected. Initially, we were doing about 1,000 for the training population every year. I've since backed that down to 600 per year go to the field to increase that training population. Relative to other genomic selection programs, our training population is enormous. The number of individuals we have in it, I think that's part of the reason it's going well, is that we have a really large training population. The sequencing we're doing is a GBS approach, where initially we had around 15,000 markers, and now recent years it's gone up to about 25,000 markers that we use in developing the equation. So how's it going? Several publications have come out over the years here where we've been tracking our progress, and I'll show you one kind of figure here that talks about the relative efficiency. This is for just yield per spike, which is, as I kind of mentioned, is one of the most important traits. This just came from the first generation with the cross validation on that from 1 year. Or it's actually 2 years of data, projecting how good of progress or how rapid of progress could we make if that was indicative of our progress over multiple years. Jerry Crane, the postdoc in this project, put together this nice graph of this is relative intensity. So actually, as I mentioned, I was selecting with phenotypic selection on tens of thousands of plants. And we can't afford to do quite that many genotype plants yet at this point. So our intensity is reducing down to like 0.9 instead of the same amount of intensity. But we can imagine if genotyping is really cheap, we could go even higher on intensity. And then the time per cycle. So one would be sticking with what we've been doing, which is one cycle every 2 years. And so even with at this sort of the point what we're at right now, we could expect somewhere around a doubling of efficiency. But if we could so that's one cycle every 2 years, but where we're at is one cycle per year. So we're into 2 to 4 times the rate of progress, a relative gain relative to phenotypic selection, just because if we can cut a whole year off, we're going to do a lot better. So what I'm excited about is that we've got a model now that's based on 3 or 4 years of data from the field instead of just that single year of that we were doing with phenotypic evaluation. So I think our evaluations are a lot more accurate, in fact, and we're doing it in just 1 year. So this is looking at a number of different traits. So this is the kind of the cross-validation prediction from 2 years of data. And you see like things like free threshing that are pretty consistent year to year. The cross-validation is really good. Seed mass, I mentioned, are pretty good. The heritability wasn't that great in that study, interestingly. I usually see it higher than that. Shattering and spike yield. And so for all of these, we're predicting that this is the relative rate of progress to phenotypic selection, more than doubling for all of them. So this is where we were at, looking at it from the kind of first selection cycle, looking forward, saying, how's it going to go? And it looked like it was going to be promising, and we continued, as I mentioned, with more years of this same approach. So I had some evaluation. Again, this is like field data in a realistic environment, crop field type solid seeding environment. And I had my first 5 cycles of selection here, and then products of the second cycle of genomic selection. I pulled out some plants that it's a little more diverse because they were selected in different ways, but this is essentially represents this is the first cycle of genomic selection and then 1 more. So this is 8 generations into the breeding program, with these last 2 being genomic selection. And we see for the traits that we're interested in, grain yield, shattering, seed mass, and naked seed or free threshing. Basically, we stayed on the same trajectory over time, or even maybe improved a little bit with the genomic selection per selection cycle. The important part is that these selection cycles took half as long. So that's why we're hoping for about a doubling of progress with the genomic selection. It'd be even better, I can mention, if we could get it down to 2 cycles a year in genomic selection, but that's another story. We'll see if we can do that someday. Another way to look at this is to look at the predicted values of the plants. So using the plots from the model, using our most recent model from all the data from many years and thousands of plants, going back and predicting the plants that were in every selection cycle, we can kind of look at our predicted improvement over time. So cycle 6 is the first or the last cycle of phenotypic selection. And then all of these are the genomic selection cycles over time. So this is spike yield in grams, which is what we hope is correlated to yield of the whole plots in the field. And we put a linear regression on here, but it sure looks to me like it's better than linear. That as our model improved over time, it's not surprising that the model predicts these to be better, because this is the model that we use to select these, whereas this is the old model back here. So that model improved over time quite a bit, especially because yield is so variable from year to year. Seed mass was not near as variable. I think our model was pretty good from the get-go. Percent free threshing, nice linear response on that one, and shattering declining in a very nice linear way. And we kind of see like, so shattering, by the time we get down to 1, that's basically a rating of 0 shattering. So we're not that far away from getting a population that's pretty consistently 0 shattering. Free threshing, similarly, by the time we hit 100, we're going to be there. So another 5 generations, and we're going to be very close to having a population that's 100% free threshing. We can obviously pull out plants to develop varieties, which is what I'm doing right now. So pulling out the best 5 individuals here that are 100% free threshing and 0 shattering and the biggest seed. And I'm using those to create synthetic varieties that it doesn't maintain all the diversity here that we have with 100 plants selected every time, but just pull out 5 and make a really good variety for farmers to use right now is the approach I'm using. And those are kind of dead ends because they're reduced diversity, but we keep this going, and over time the whole population moves along. We hopefully keep that genetic diversity that allows us to go for a long time. Okay. I have to wake up and remember why I put this here. Oh yeah. So additionally, how we might be able to accelerate is if we could find QTL that are associated with some of these traits. Maybe we could add those in as fixed effects in our selection model. Maybe we'll do better than just a genomic selection model based on average values. There's some evidence that works for other species. So we had we've had several QTL studies done at the Lansing and collaborators at other places. So our grad student Kayla, who led this project, and it was did association mapping as well as a NAM, or a nested association model. So there was interval mapping as well as association mapping, and found some outstanding QTLs that were very significant for traits. This is like free threshing ability or nakedness of seed that jumped out, were really nice. And as we look though at like how consistent they were across locations, in order to make it seeable, there was 4 locations in years. I just zoomed in on one and many traits. So here's just a little piece of the table looking at the effect of the alleles. And so the effects are here, and for brittle rachis, and here is the percent of variation explained. So we see even if they're significant, they're generally low, less than 5%, and then they're hit and miss from year to year. So generally what's significant in one year isn't the next. So even for a trait that's highly heritable like brittle rachis, we see that even the best ones are not, QTLs are explaining less than 5% of the variation. Here for shattering, we had, here's one that was huge and explaining 50%, but then at the other locations, very low. So frustrating to get just single QTLs that are a slam dunk that would allow us to really rapidly improve any of these traits. And yield is of course even worse. So we've been frustrated basically deciding that the genetic architecture is it's kind of the infinitesimal model of a huge number of alleles controlling these traits, each one having a very small effect. And so the genomic selection model, just using a linear model, it works well. Might there be another approach by using what we have from wheat? And as I mentioned, my work here started with wheat-whited hybridization, and we have plants now that are crosses between durum wheat and intermediate wheatgrass that have all the chromosomes of intermediate wheatgrass with all of the durum wheat chromosomes added, and then that's been doubled. And so I looked at back crossing those to wheatgrass, using intermediate wheatgrass as the female. That's extremely difficult. Lots of embryo rescuing, got about 3 plants out of that, thousands of crosses. And those had to be embryo rescued, as I mentioned. And then the next generation, trying to cross those back to wheat again. You can see I was excited to find 1 seed, so just a handful of seeds on again a bunch of spikes crossed to wheatgrass. Trying to get back to intermediate wheatgrass with just a small amount of wheat. And we ended up using GBS once we had a large number of plants that have been back crossed into 1 more generation. So these are all the wheatgrass chromosome. This is the GBS read count from the aligning to wheatgrass genome. You see all of the chromosomes of wheatgrass are there, and then these are the durum wheat chromosomes that are present. So we had an inexpensive way to start looking for the presence of wheat chromosomes in these plants. And here's an example of what I really want to find. The plants that have a mishmash of wheat chromosomes tend to be, well, they're going to be very unstable, and they're also tend to not look very good. Once we get down to one chromosome, they look just pretty much like wheatgrass again. They look healthy and normal, but maybe they don't have much improved from wild wheatgrass. We don't know yet. So my next project was to try to obtain a whole array of single chromosome additions into wheatgrass. And I believe I achieved that pretty much a year ago. And we now have a study out in the field where we have what we hope are all of them. So we now have done a new approach of skim sequencing them, working with Jesse to develop this method. And this is a zoom in. So you see here at the top are the wheatgrass chromosomes down to 21 wheatgrass chromosomes. And beyond this, we start with the wheat chromosomes. And so this particular plant, yep, it was what we want. It has just chromosome 3B added in to the intermediate wheatgrass. So we've got a little over 1,000 plants in the field now that we're doing the skim sequencing again to confirm that they all are what we hope. Certainly some of them won't be what we hope. There's going to be a few things that don't fit what we're hoping to find, but for the most part, we'll be able to say, yes, these are single chromosome additions, and then look at the phenotypes as they're influenced by those chromosome additions, as well as using the markers, hopefully, to take out the wheatgrass background, basically looking at it up just based on relatedness between these wheatgrass plants, and then adding in the effect of this wheat chromosome. So exciting new project to see if we can make some leaps forward in a few traits such as short stature or larger seed. Finally, the exciting idea of using targeted mutations in intermediate wheatgrass to make rapid domestication. There's been a whole handful of papers that have come out suggesting we could rapidly domesticate tomatoes starting over, because we know all the genes for domestication. And so is it possible to take what we know about wheat and barley and rice and bring that into intermediate wheatgrass by just editing the genes to be a domesticated form, essentially using CRISPR to do that? Is it possible, or mutagenesis to achieve that? So I'd love to get semi-dwarf intermediate wheatgrass. The high yielding plants are always tall, and it's frustrating. The short ones are just crappy. So would it be possible if we know the dwarfing genes, to edit these wheatgrass genes to suddenly get dwarfing? So we had a paper that came out of some funding that Michael Palmgren got for work in Denmark and his team to look at the wheatgrass genome for candidate genes that could be edited. And so this is just a table, a piece of a table from that publication, showing that, yes, there's often 3 loci, at least in some cases more like 10, that unfortunately that match to things like brittle rachis, known domestication genes. And these could be knocked out, or in some cases of Q, perhaps, to make the actual edit that we hope will result in the phenotype that's similar to the domesticated form. So it'd be super exciting if we could bring that super domestication gene of Q that was so important in the domestication of wheat, if that same change could be made in perennial relatives and have a quickly domesticated type form that would have larger seed, shorter heads, free threshing ability, and non-shattering, which is what Q presumably did in wheat. So, and then many other traits. But some of them, as I was mentioning before we started, I think that if you look at sort of the shape of wheat, and all of wheat sort of looks like this, and we don't really know the wild wheats were more long and narrow, and what made this change. Like that gene hasn't been cloned because it's not in wheat anymore. And so being able to know that the gene that made that change would allow us to hopefully make that similar seed shape change in intermediate wheatgrass, as we've as I've bred for doubling or tripling of seed size, they just keep on getting longer and skinnier. And we really need to find some way to make them plump, and that genetic diversity doesn't seem to be present in the species very easily. So it's one that's really would really be great to use as a target from on this approach. So yeah, I hope I've covered sort of 20 years of my work in an hour here, and there's so much that we could go into in many different directions, and I'm sure you have lots of questions about what we're doing and why we did it. Happy to talk about it, and I'm convinced that the future as we bring these new technologies is just gonna we're gonna accelerate even more, and it's gonna be feasible for the first time to start looking at developing new crops from scratch in not 50 years or 100 years as we used to say, but hopefully in a decade or two.
So, awesome. Thank you, Lee. I'll clap. Good, great stuff. Yeah, we've been working on that wheat to intermedium integration, and it just occurred to me that's like opposite of all of the wild integration work.
Yeah, exactly.
All of the wheat breeders have been doing for decades and decades. So it's a interesting irony, I guess. So, okay, let me open up to any questions. Feel free, please unmute yourself or raise your hand or type in. Nico, I got a first question there. Go ahead.
Yeah, thank you. What an inspiring talk. It does seem like it requires a lot of patience. I have several questions, but I'll start with increasing weight of the seeds and maybe number. I don't know. Do you run into problems where the stem can't keep up and you get lodging, or they fall over, they bend over? That makes it harder to harvest, stuff like that.
Yes, definitely. So that's actually been one of the traits I've been selecting on. I didn't happen to mention it here, but yeah, ever since we did genomic selection, I've added it in because we can evaluate those plants in the field for several years. The lodging often happens in the second, third years. The first year they don't tend to lodge. The plants are shorter and the stems are just more strong. That crown, as it ages, seems to weaken. We've thought that reduced height would be the key to this, but interestingly, as I measure height and lodging, the shortest plants are lodged the worst, and that's probably just due to a correlation between general weakness of the plant and a crown that's becoming weak, and small and spindly stems. So the big, tall, thick stems have a lot of strength, and they tend to have a lot of strength at the base, so that the lodging isn't the stems breaking. It's what you might call crown lodging, and so it's a heavy rainfall where it softens the soil around the plant, and then you get a little bit of wind and it will fall over. So parts of the country that don't get heavy storms in the summer don't have much lodging, but definitely it's a trait that I care about a lot and giving a lot of attention to. And I would like to have a really strong stem and a short plant, but that's been the frustrating piece. And hopefully some of these other approaches, genome editing or wide hybridization, we can get dwarfing genes in that will, a dominant dwarfing gene, say from wheat, that might reduce the height of the plant without making it into a wimpy plant. So yeah, it's super important.
Right, yeah, that's actually what I would like to ask a question about as well. You say you would like to go towards a more dwarf-like phenotype, but
Yeah.
Are you keeping an eye on the root biomass? Because can a smaller plant sustain that? Yeah, I guess it's a lot of work if you want to look at root biomass, because it goes so deep.
Right.
But this is one of the reasons you selected this species to work with in the beginning. So losing that would be bad. Is that monitored or?
Yeah. Every time I'm in a group with agronomists or soil scientists, I always say, my breeding plots are open to you, and I invite you to take soil cores out of 100 plants and work with me on this. I want someone to do that, and so far no one's done it. It's frustrating, but there's a large project that's been funded in the US. It's called the CAP funding. It's about $10 million from the government for a 5-year period. And part of that work does involve looking at root biomass under some different systems and things. So I think there will be some work done, and I think we're going to go more there in the future. To broaden the topic even more, so yeah, we know that dwarfing genes in wheat have had some correlated reductions in root length, and so we might expect them to do the same thing here. In terms of sort of overall root biomass, asking how much would be reduced through breeding. But there's a grad student who just did a modeling study on root biomass and sort of an ecological evolutionary approach to selecting for grain. And her model suggested that we might give up 10, 20% of the root biomass to get a trade-off with grain yield that might happen. For the most part, we'd be pretty okay with that because the root biomass is so large already. So if we're going to no tillage and we still have something that's got, instead of having 5 times the root biomass of wheat, it has 4 times, it's still enormous. And whether that really matters in terms of the ecosystem services, we don't know. But those are all things that we propose studying. But we're pretty sure that if we go to a system we're tilling maybe once every 6 years, so we don't suppose the sand's going to last forever, but 4 to 6 years is a good target. So we might only be disrupting that system once every 6 years instead of annually, and then having a root biomass that's 2 to 4 times greater than our annual crops is still enough. So we're not seeing a small sacrifice of root biomass as being a game changer or breaker for us. Interestingly, the USDA government funding agency put out a proposal for trying to increase root biomass in annual grains as a way to increase soil carbon sequestration. And we are wanted to apply to this and said, well, look, we've got it already. We've got the root biomass. We just need to get the grain. And they weren't interested in funding that approach. They only wanted to try to get a 5% increase in root biomass of corn or something. So we think it's a lot harder to increase root biomass of maize by 5% than it would be to get more grain by decreasing our root biomass by a small fraction.
Great questions. Good point, Lee. I'm still annoyed about that RFP. Vanessa's got a question. We'll go there next and then to Brandy after Vanessa.
Okay. Hi, Lee. My video will start soon. Thank you. That's a really interesting presentation. And, a lot of perennial legumes are being used in Australia on marginal lands, and it'd be great to see this wheatgrass coming into effect as well. Okay, I'm on. So I'm interested in your recurrent selection, and particularly, it seems quite a laborious process to be selecting for shattering, seed size, and free threshing ability. Have you come up with any miraculous new methods to speed this up in your 5-year breeding cycle?
Yeah, like the original phenotypic selection.
Yeah, the original. Yeah.
Yeah. Yeah, in some ways, genomic selection is how we figured out to speed it up, and that's a big piece of it. But I've also learned that I didn't have to cut near as many heads per plant, for instance. So we were initially cutting large numbers of heads and threshing all of those. And I discovered that something like seed size and shattering and free threshing, especially, and even for yield per head, 3 heads was enough. So some of those generations, we went down to just, so you see those plants had like 100 to 500 heads per plant.
Yeah.
And we just took 3. We did it non-randomly. So if we took random heads, it's not very good. But if you take heads that were consistently some of the largest ones on the plant, that was much more heritable. And so we're so ingrained in us to use randomness all the time. In scientific studies, we always want to take a random sample, but I found that by not taking, so if I wanted to be a random sample, I needed like 50 heads, but if I took a non-random sample, it was very predictive to take only 3. So that allowed us to have 20,000, and I used like long bread bags and put 3 heads in each one, that we could, it was actually feasible to harvest 20,000 plants and bring them all back. And it wasn't this mountain of biomass. Initially when I did 1,000 plants and I took like the whole plant, it was a mountain of biomass that the mice were getting into by the time we get it threshed, right? So, so just sampling very small numbers of plant, or very small numbers of heads from each plant, when you realize that what you're selecting on is so highly heritable and there's so much diversity at that stage, you're looking for the needle in the haystack. And I didn't put up the distribution of these traits, but a lot of these traits were really highly skewed distributions. So we're looking for the plant that's on that long tail end of the distribution. So that the average yield per head was like a 1/10 of a gram, but the ones that I found to select were 3 times that. They were 0.3 grams. So the selected fraction was like 3 times the average. So that, indeed, if you would have gone and taken all the heads of the plant, you would have found, well, it probably wasn't quite that good, but those are really, really good plants. And what I found was that when we looked at the distribution of those selected plants, say if I selected 100 and then I reevaluated those, the top 5 of those plants were really outstanding versus the rest of the other 95 as well. So we were finding these few extreme plants that then I tracked the pedigree over time, and I found that quickly about 5 plants, their progeny took over this entire breeding program.
Yes, that's what I'm interested in. Yeah.
So I used a pedigree approach and tracked. Not initially, not the first generation or 2, but eventually I was using a pedigree approach where I tracked the mothers and fathers of it. Actually, only the mothers. We were just using a outcrossing population. So I knew the mothers of every plant and saw that these few really outstanding plants were really taking over. And it's kind of surprising, because you think, well, if you're going to be able to domesticate something, you should be able to find that trait in the population. And I went through thousands of plants for several generations and never found a free threshing, non-shattering plant. We just selected the most free threshing, non-shattering plants. And then suddenly, in my 3rd generation, I found one plant that was completely free threshing and completely non-shattering. And I thought, whoa, it was the one gene, like this must be the Q gene, had some mutation, and we found it. And so then I started crossing with that plant. I thought, this is going to be very easy. Wow, it's one gene. And then with QTL studies, we found, no, it wasn't. It was just, after repeated cycles of selection, we finally accumulated enough genes to get one plant that had all the pieces come together to something that suddenly jumped out as, wow, it's free threshing, non-shattering. It's like a domestic plant. And there it was. And then it took like 5 more generations to recover something that was as good as that plant again, because it was very quantitative. And we just had to keep on selecting for those traits until we get back to enough alleles accumulated. So it's interesting that we know that major domestication genes and these QTL studies that I didn't go into in detail have a lot of hits that are right next to known domestication genes, but then there's another 15 hits for the same trait that's not in normal places. And who knows what was initially part of wheat domestication even. So there may be a lot of things that we've lost that were important. And we find, okay, this, we take one gene out of modern wheat and we can clone it into wild wheat and we get instant domestication, or we could put the wild one into wheat and say, oh, this one gene is so important, but maybe it's actually the background as well. So who knows? The story of domestication, there's a lot of papers out there, and I'm curious about all of them and read them, but they're always just like sleuthing around in the history of 10,000 years ago, trying to figure out what happened. And what's weird is like we're doing it for the first time. We're starting over with modern tools to figure out what happens. And
Yep.
maybe these perennial grasses are really different, but maybe it's a lot like what happened with wheat. I don't know.
Thank you. It's really good advice for us as we embark on some neo-domestication projects of our own. So thank you.
But
Good. Great question, Vanessa. Thanks. Randy.
Yeah, thanks for that, Lee. That was very impressive. I'm so focused on gene cloning and genomics, so it was really nice to see this tour de force and how fast you can go with conventional breeding combined with genomic selection. So building on from Vanessa's question and the challenges you had with finding the free threshing trait, do you see a sort of genetic bottleneck of diversity in your breeding program? Has the genetic diversity gone down over the years with successive cycles? And so I'm a disease guy. I work on disease resistance. So I was wondering, did you over the years see any shifts in the pathogen population and increased this incidence of disease?
Yeah, so first the diversity question. Yeah, we certainly hope we're reducing the diversity to some extent, because that's the only way we're going to make progress. But that's always the plant breeder's conundrum, right? So we have to give some up in order to make progress. But are we undermining our future progress and losing things that we wish we weren't? And so with the genome, we've been trying to find some nice stories, which would be like a signature of domestication or selective sweeps in the breeding program. Some slam dunk, okay, we fixed this haplotype, and it's been, Jesse can chime in, but it's been frustratingly difficult to find any region that we fixed. In fact, every region still has diversity. There's nothing that we fixed, I think, in the breeding program yet. So that all of these things remain fairly quantitative, and that it's because there's many ways to get the same phenotype, I guess, that we're not fixing one single haplotype. Definitely we're losing diversity to some extent, but Jesse, maybe you could tell your conclusion of when we looked at the number of haplotypes or the number of gene variants in like 3 plants, we counted up how many there were relative to the Summit wheat breeding program.
Yeah. So Lee's quote unquote like small breeding program of a few thousand plants, 100 parents each generation, has got something on the order of the same level of diversity as the entire spring wheat gene pool or something like this, at least as much as the entire Summit wheat program, some of these really, really diverse ones. And we think that's largely just because, this is just a few generations removed from a completely wild population. And also because of the breeding method, this has been going through, right? These are not inbred and like single parent selection. These are like multi-parent populations that are just slowly moving allele frequency. So yeah, we see in those few plants, you see as much haplotype diversity as you do like in all of the domesticated wheat. Yeah, so yeah, we joke that Lee's problem is almost like you've got too much diversity and needs to get some of it out.
Yeah, it might simplify things. In fact, if I went down to selecting 5 plants for a couple of generations instead of 100 and try to cut through some of this diversity, but for the long term, that's why my approach is, as I mentioned, is to create these varieties that are only 5 plants, so we can skim off some very best material and make a variety, but then keep that diverse gene pool. We've try to look at, in these generations of the genomic selection, we can look at the change in gene diversity over time in there. And it's been maybe a little bit, but it's almost undetectable, the decrease in genetic diversity in those populations in 5 years of 4 years of genomic selection. In terms of disease, the plant here is mostly immune to the major diseases of wheat, so rusts and things. The diseases we do have are ergot, and that seems to be mostly based on the kind of year we have. If there's poor pollination or a lot of rainfall at the time of pollination, you can get ergot infestation. Instead of breeding that out, we're basically people who get contaminated seed use a color sorter and take it out that way. That's like the simplest solution right now. Bacterial leaf streak is a serious disease that happens when it's hot and humid, and there are some plants that are highly susceptible to that. We're trying to include it a little bit in the genomic selection model, so we're including some genotyping for that. But the highly susceptible plants, well, it hasn't been associated. Like if we rate it in the field, it doesn't correlate with the yield at all or any other trait. Part of the issue is that these plants, by the time that they are heading, they've got a lot of biomass, and it seems like the fixation in the head is all they need. They don't even, because you have this huge head with relatively small seeds in it. The leaf material being lost doesn't really seem to matter. So loss of leaves to that disease doesn't impact us much, but it does impact the forage quality. And so farmers are going to care about it for that reason, and it looks terrible. And it can move into the head to some extent as well. Scab is the one that's probably the biggest concern, because we can get loss of quality and able to use the seed due to contamination of vomitoxin.
With scably, have you checked to see if your population contains FHP7? Because that was cloned from Thinopyrum. I think it was Thinopyrum elongatum, but I think it's also found in some of the other Thinopyrums.
That's a good question. I don't know the answer to that. I have not looked, but there, as we have done a fair bit of screening of plants in the breeding program for Fusarium, and some plants are completely resistant, and the average plant is as good as basically the most resistant wheat that's available. So it's quite resistant, but nonetheless some plants can be infected. And so really bad years, farmers have had fields where the level of vomitoxin has exceeded what would be considered safe. So only a few instances so all of the grain gets tested. We don't sell any, you can't go to the market. It's our policy that has to be tested. And there's only been a few instances where it has been too high. And that's growing in places that are really bad for Fusarium in some situations. So, but breeding for resistance to it is important to us. It's been tricky because the year-to-year variance has been really high. So we've been trying to make a genomic selection model, and unfortunately we can't even predict the next year yet. So it looks like we're going to have a lot of years of data to get a good model. But maybe as you mentioned, it'd be worth trying to just genotype for some known resistance genes already, and see if they have an effect. That's a good suggestion.
Okay, thanks a lot, Lee. Very, very inspiring, very interesting.
Thanks, Brendan.